Hi, I'm Delaney Fisher from the Price Lab at UVA. I'll be sharing my work on a computational biotransport model of the glymphatic system, wherein we predict that focused ultrasound can increase solute clearance. Let's begin by reviewing the glymphatic system. The glymphatic system is a relatively newly identified system that exists in the brain to enhance clearance of metabolites as the brain lacks the classically described lymphatic system. The glymphatic system pulls cerebral spinal fluid from the subarachnoid space that surrounds the brain and the brain into the brain tissue along penetrating arteries in what is termed the periarterial spaces. The space is lined with astrocytes, which allow the cerebral spinal fluid to enter the brain through aquaporin pore channels contained on the astrocytic end feet. The cerebral spinal fluid traverses across the brain parenchyma to astrocytes lining the perivenous spaces, where the cerebral spinal fluid will then return to the subarachnoid space and eventually drain through the venous sinuses or meningeal lymphatics. The cerebral spinal fluid flow conducted by the glymphatic system allows for convective flow to enhance the clearance of brain metabolites and other brain solutes from the parenchyma, which would otherwise be a very slow process through diffusion alone. While we are quickly learning more and more about the glymphatic system, we still understand very little about how FUS and FUS-mediated therapies affect the glymphatic system. A recent study by Meng et al. Has indicated that focused ultrasound application to the brain in humans can enhance brain solute efflux. Another study by Hirsch et al. Found that pulse ultrasound can increase the size of perivascular spaces in ex vivo brain slices. To better understand the role of FUS on the glymphatic system, I created a basic fluid dynamic model of the glymphatic system and analyzed perivascular spaces in histological images of FUS-treated brains to recapitulate an effect of FUS on the glymphatic system in silico. This 2D model was produced in COMSOL and is comprised of 5 compartments that represent the 2 perivascular spaces, the astrocytic end feet or gliolimitins, and the brain parenchyma. The perivascular spaces are modeled as fully developed laminar Stokes flow, while the gliolimitins and the parenchyma are simulated through the Brinkman equation as a flow in porous media. To model solute transport, parameters were used for a solute equivalent to 70 kilodalton dextrin, which is commonly used as a tracer molecule, at an initial bolus concentration of 0.5 micromolar within the middle third of the parenchyma. To understand one effect of FUS on the glymphatic system, histological images from FUS-treated and sham-treated mice were analyzed for the perivascular space diameter as a ratio to the vessel diameter. In agreement with Hirsch et al., our analysis revealed that FUS-mediated blood-brain barrier opening resulted in an increase in the perivascular space diameter to vessel diameter ratio by 26%. These results were reflected in the model by increasing the periarterial and perivenous space widths by 26% each. Comparing the temporal profiles of solute clearance for FUS negative and FUS positive groups indicate a faster solute clearance in the FUS treated group. The increased perivascular space widths augmented the volumetric fluid flow rate in the periarterial gliolimitins by 10%, the parenchyma by 9%, and the perivenous gliolimitins by 12%. Correspondingly, the FUS positive simulation resulted in a 12% decrease in solute half-life in the parenchyma compared to simulations with FUS negative perivascular space widths. Ultimately, our model of the glymphatic system indicates that FUS-induced enlargement of perivascular spaces is predicted to augment solute clearance from the brain parenchyma. It's important to note, though, that this work currently only explores one possible mechanism of FUS-induced effects on the glymphatic system, but it's likely that many other effects of FUS on the brain contribute to enhancing solute efflux. Moving forward, this and future models of the glymphatic system can provide insightful predictions for many applications of FUS treatment, such as temporal concentrations of drug and gene delivery, amyloid beta clearance in Alzheimer's disease, or even tumor antigen clearance for immune cell priming. And before closing, I'd like to thank my advisor, Rich Price, and my lab mates for their support, and the generous funding agencies that support our work, as well as thanking you for your attention.